So I'm really excited to have today Dr. Uri Eden, and he's a professor in math and statistics at Boston University, and he's the director of the BU Statistics program. And I think we're very lucky to have essentially probably one of the world's experts in the field that he's about to talk about, is application of point process analyses. And his background is he got his BS in mathematics and engineering and applied science from Caltech. And then he got his PhD from Harvard in Harvard University, and then a postdoc fellow in the BCS department at MIT. And so now he's part of the math and stats program in Boston University, which is actually where I did my PhD thesis. And his work focuses on developing mathematical and statistical methods to analyze neural spiking activity and integrating methods related to model identification, statistical inference, and signal processing, and stochastic information and control. And so I've been definitely plugging this, that he's also a co-author of case studies in neural signal processing, along with Dr. Mark Kramer, which is really just a fantastic book that I have everybody that joined my group read. It has a wonderful overview of everything that you really need to know if you're starting out in signal processing. So with that, I'm going to turn it over to Uri, who is going to share some really exciting stuff with us today. Let me switch this over.
Great, thank you very much, Dr. Perrow. Can everyone hear me?
There you go, and then, okay, I think we're all set. And
Okay.
actually, one thing I wanted to add before we started, so if you see on your bottom panel, there's a Q&A button. If you put questions there, Dr. Eden can either field them directly, or at the end, maybe we can go through and we can go through and answer some of those questions. Okay.
Great, thank you very much. So as Dr. Perrow mentioned, my research is focused a lot on analyzing neural spiking data and using point process models to analyze that. And recently we've been branching out, and I've been working with Dr. Perrow to try to understand how the same type of point process models can be used to understand event data in sleep. And so I was going to present today just a little bit about the basics about how to understand event data using point process models, and some of the ideas we've had about using these types of methods to analyze sleep data. So just a brief outline, I'll start just by sharing a few things, a few areas where point process modeling is important, and some of the questions that arise, and some of the real basics about how to look at point process data, some of the basic statistical methods, and some of the basic models. And then I'll pivot towards just a few small examples from neural data analysis, and how point process modeling has been used to really understand things that wouldn't be identifiable just through standard statistical methods. And we're going to take those lessons and then try to apply them to data from sleep disordered breathing, in particular trying to look at an instantaneous measure of the event of the rate of respiratory events. So let me start by just defining what a point process model is. So a point process model is a type of probability model that's used to describe any physical system that produces data, random data, that's comprised of localized events in time or space. And so these are useful anytime your data can be described as events that are located at a certain point in space or at a certain point in time. So that's kind of the way to describe what point processes are, but maybe a visual way can communicate, to my mind, better what we actually mean. So oftentimes when we're dealing with, say, time series data, if you look at something like the temperature through time or stock prices, you'll see a continuous process where at every moment you might get a different possible value, and those values are changing moment to moment. On the other hand, event data or point process data is in many ways much simpler. It still occurs continuously in time, and you just have localized points in time where events are occurring. And so fundamentally, there's a kind of simplification here. At any moment, you either have an event or you don't. You have a binary outcome. So in many ways, dealing with point process data should be simpler than continuous process data. That said, a lot of people's statistical training starts with the idea that data is continuous. It comes from a continuous distribution, like a normal distribution. And so many of those methods don't apply well when the data are more localized event data. So I'm going to go through some examples of certain fields where point process data analysis has really caught on and led to some really great discoveries. And one of the areas where a lot of the theory related to applying point process models to the real world comes from is seismic data, the analysis of earthquakes. So earthquakes are localized both in space and in time. In space, you can localize, say, where the origin, where the epicenters are spatially over the globe and in terms of depth. And in time, you can localize when exactly these events are occurring. And so once again, these are localized events. They're occurring at a particular location and at particular moments in time. And we can think about the types of questions you might be interested in regarding these seismic events. In particular, the prediction question is of fundamental value. So we're often interested not just in characterizing and building statistical descriptions of the data, but in predicting when the next event will occur, where it will occur, and features of those events. And so prediction is essential. But as well as prediction, we're also interested in things like association. So what things are related to the events that occur. We're interested in patterns of events, so things like aftershocks or periods of quiescence preceding major events. And we're also interested in understanding mechanisms. So by looking at when events occur, can you estimate something about the unobserved latent processes that are driving these events to occur? And so for the past 50 years or so, there's been major advances in applying point process models to this type of data. Another area where point process modeling has been quite useful, especially recently, has been in the analysis of cardiac data. And if we look at human heartbeats, certainly there are features in, say, EKGs, continuous features that are important, things like the shapes of various complexes here. But fundamentally, you can also think of these heartbeats as localized events in time. So when, say, each beat is occurring. And in a case like this, we're much less interested in prediction. It doesn't really matter if we can predict with high accuracy when the next event is going to happen. Oftentimes here, we're much more interested in matters of association. That is, can you look at, say, the variability of these heartbeat events, and by relating the variability to other variables, can you understand whether or not a subject is healthy, whether or not they have certain disease processes? Can you predict something about the prognosis for those processes? And we may also be interested in a case like this in underlying mechanisms generating these heartbeats and whether or not those represent healthy processes or disease processes. Another area I'll mention is nuclear medicine. So in a PET scan, for example, a clinician will introduce some sort of radioisotope incorporated into some metabolically activated molecule. And those molecules will gather in certain tissues. And then when you have a radioactive decay event, those events are occurring at localized points in space, in the tissue, and in time. But that's a point process that's not directly observable. Instead, you have a set of photon detectors surrounding the tissue. And what you do observe is a set of point process in each of those detectors. And you're trying to use the observed point processes, those detected photons from each of those detectors, to infer some kind of unobserved latent process, which is the decay of these radioactive molecules in the tissue, where and when they occurred, and use that to try to reconstruct an image related to those tissues. And so again, you're probably less interested here in predicting when events will occur in each one of these sensors, and instead interested in what the events that occur at those sensors tell you about the system itself, and fundamentally how certain you can be about that. So if you see maybe some reduced activity or some reduced amount of radioactive events in a certain area, can you tell whether that represents a statistically significant reduction and what that might mean for a particular disease process? And then the area that I've spent much of my time looking at, neural data analysis. In the past, I'd say 20 years or so, there have been tremendous strides applying point process models to neural data. And here the relationship maybe is fairly obvious. You have neurons in the brain or neural tissue in the rest of the body that is responsive to various biological or behavioral signals. In this case, we have a stretch receptor in a muscle, and depending on how much you stretch the muscle, you get different amounts of activity. If you are able to put an electrode directly into the cell, then you may be able to observe sub-threshold membrane potentials that might relate to the stimulus driving the neuron. But mainly, we're able to put electrodes nearby these neurons. And in those cases, we're not able to observe these continuous valued signals. Instead, we're just able to observe these discrete events in time. And so here again, we're mostly interested in understanding relationships. How does the quantity of these spike events translate? How does it relate to the biological and behavioral signals driving things? Can you build coding models that help you understand these relationships? And then can you use those coding models to predict something about the either cognitive processes or biological signals that are driving neural processes? And then, as I mentioned, we're trying to move towards applying these same classes of models to various types of sleep data, in particular to respiratory events. And in particular, you might think of all sorts of different types of time series data you might collect during a sleep study. You may have EEG activity, which mainly looks like it's continuous valued. At every moment, you record some potential that's changing. But you may think about discrete localized events that are occurring in that EEG data, things like spindles, or you could infer things from that, such as transitions between sleep stages. In our case, we're going to be very interested in discrete event that represents either the times of onset or offset of respiratory events. So periods where, say, the airflow is stopping or starts back up again, we can treat those as localized events in time. And again, we can try to ask questions about prediction. Can we figure out when the next event's going to be? About association, what types of patterns of events occur and what are they related to? And things about maybe even mechanisms, what cognitive and physical processes are leading to each of these events. So those are kind of the types of areas that we've been thinking about. I want to spend a few minutes just talking about the kind of fundamental idea, the kind of common types of questions and methods we use to analyze those questions. And as I've pointed out in these examples, I think there's kind of three categories of questions that arise quite a bit when analyzing event data. The first that I mentioned is prediction. So can you predict, and how confident can you be about when the next event is going to occur? And that's really a sense of how random the events are or how predictable they are. We have often questions of associations where we record this event data alongside other variables that are observable, and we want to understand the relationships between them. We want to understand, for example, the relationships between events and past events to understand what types of patterns of events occur. And we might want to understand how events are related to maybe other features of the neural activity recorded in the EEG. And we're often interested in mechanisms, that is, trying to infer underlying unobserved factors that are actually generating or driving the processes that lead to those events. And maybe trying to understand how the dynamical properties of those underlying factors lead to the types of patterns and dynamics that we see in the event data. Okay, so those are the types of questions we're often interested in. How do we first start thinking about point process data? Well, one thing that's both a challenge and an opportunity with point process data is there's often multiple ways to think about the same data, as opposed to maybe a continuous process where we're just looking at the value of the process moment by moment. when you're dealing with a point process, you can record the data in multiple ways, and you can think about building probability models in multiple ways. For example, you might record just the times of the events and try to build models for those times. Or you might look at the waiting times between individual events, and you might be able to construct particular class of distributions that capture those waiting times well. Or another thing that people often do is they try to build a process that describes the number of events that have occurred up to and including a particular time t. That's typically called the counting process, which will write n of t, where n represents the count of events at time t. Or sometimes you can break those up into smaller time bins and you can just ask how many events are occurring moment to moment. And can we characterize a model that characterizes the probability of seeing an event moment by moment? And a key feature there is if we bin time in small enough bins, then hopefully we can get to a point where at every moment either we do or we don't see an event. And then we can use simple binary probability models to describe that data. It turns out, and it's fairly easy to show, that there's a one-to-one relationship between all of these ways of describing the data. And so if you build a probability model for one of these forms of data, you get for free tools to fit and assess the quality of the models for any of these other forms of data as well. Okay, so that's one way of characterizing the data. How do we actually build a probability model for the data? One way we can maybe think about this is as an engineer might, as a black box that has some inputs and outputs. We have as our output a time series of these events when they occur. If this is stochastic, then it may be random what the particular time series is. If I repeat, say, the same stimulus to a neuron, I might get a different pattern of spikes each time. But there's going to be some probability distribution that characterizes what the probability of that particular pattern of events is as a function of all the various inputs that may be affecting things. And those inputs could be continuous valued signals. They could also be point processes from another system. You may have a network of point processes that are interacting. Or they may be some model parameters that are either fixed or changing in time that you may be interested in estimating. Now, it's a bit challenging to write out a general probability model for any pattern of events as a function of any set of inputs. But it turns out, based on the insight that I just mentioned, that you can make small time bins in which you either see an event or don't see an event, that there's actually quite an easy way to construct these probability models. That is, you take these small time bins and you characterize the probability, yes or no, that you see an event in each one of those small time bins as a function of all the inputs up to the current time. However, there's one caveat, which is that if you want to be able to accurately describe the probability distribution of any pattern of events, you also need to feed back the past history, what I'm writing here as n from 0 to t, the past history of prior events into the model itself. And so that adds one extra input to the model. The model has as inputs everything that drives it, as well as the past history of events. And this allows us to construct probability models for any arbitrary pattern of events. And it turns out those models can be expressed mathematically using a very simple tool, which is called the conditional intensity function. That's really a fancy way of saying an instantaneous rate, a rate that's changing moment to moment. And so we'll write this lambda as a function of t and as a function of the past history as the symbol for this conditional intensity function. And what it's characterizing is the probability of seeing an event in some small interval from t to t plus delta t, given the past history of all the inputs and the past history of the previous events up to that time t. And we take that probability in the limit as the interval gets smaller and smaller and divide by the size of that interval to get an instantaneous rate. And it's fairly easy to see that for any physical process that generates discrete events in time, you can characterize the probability model for that process by defining a conditional intensity function, by defining at every moment, at that instant, the rate of events. Now, if you've taken an advanced probability or stochastic process course before, you may have learned about Poisson random variables or Poisson processes. A Poisson process is one particular type of point process, and a rather limited one that assumes that every event is independent of every other event, it turns out that this idea here generalizes that notion of a Poisson process to allow you to describe any event data. So you really go from a very limited class of models to something that can characterize any point process. And the nice thing is, once you define this function lambda, once you specify at each moment what this rate is going to be, you get for free all of the statistical tools that are used to estimate models, to make predictions, to assess the quality of a model, to refine models, et cetera. And so, for example, here it's easy to see that if you want to figure out the probability of seeing an event in a small interval, you can just multiply both sides here by delta t, and that's going to be approximately lambda, the rate, times delta t, the amount of time, gives you the probability or the expected number of events in that interval. Similarly, you can build a full likelihood of observing any pattern of events. And there's a little bit of math behind this, but it's not too difficult to write out an expression for what that likelihood is. And it's a function of your data, these increments of whether or not you saw an event in every interval, and this lambda, this instantaneous rate of events, moment by moment. Okay, so that's how we define our point process models. As I mentioned, once you define the model, you get for free all these tools that help you characterize model uncertainty and the quality of your model fit. In particular, you get tools for building confidence intervals. And those are confidence intervals about features of your model, as well as confidence intervals about any predictions that you use that you generate from your model. You get tools like analysis of deviance, which, if you're familiar with basic statistical methods, is an extension of analysis of variance, which lets you describe how much of the uncertainty or variability in the data is being fit or described by your particular model. And then there's a tool that's specific to point process data, which is Komorov-Smirnoff plots on rescaled event data, where you use the model to rescale your waiting times between events, and then you compare what your model predicts with what you actually observe, and use that as an overall measure of goodness of fit. How well does your model describe the structure in the data in total, and as a tool to figure out how to best refine your model to add components if there's a lack of fit. Okay, so I think that's everything I want to say about kind of the fundamental structure of point process models. Next, I want to take a few minutes and just go through a pair of simple examples among many that we've worked on, where neural data analysis has taught us a few lessons about the importance of applying particular classes of models and the usefulness about thinking of event data using point processes. And a reason that these methods have been particularly useful for neural data analysis, and why we think that they'll be useful in a number of other fields, including analysis of event data in sleep, is there are particular challenges in looking at event data that really defy what you learn in either elementary stats classes or even in more advanced stats classes. So for example, with neural data, your counts of spikes are often fairly small compared to what you're looking at. And so the kind of stats 101 assumption that your data comes from a normal distribution is almost always wrong. And even beyond that, the assumption that you have kind of linear relationships between the things that are predicting and the things that you're and the things that you're using them to predict, those are almost never linear. And even if you go and take more advanced stats classes, you still typically are making some assumptions that don't normally hold for neural data, and in particular spike or event data. In particular, repeated observations over, say, intervals are rarely independent. The past event history almost always tells you something about what's going to happen in the future. And related to that, event statistics are rarely stationary in time. That is, the parameters representing their distributions, their means and their variances, tend to constantly change in time. And there are a number of other challenges that come up with point process data, In particular, you have neurons that are interacting in a network, and you're often interested in the relationships between those. And one of the fundamental things is we're often not recording all of the data from a physical system. We're recording often a small subset. So most of our data is missing, and we're trying to understand relationships among the small fraction of sources that we're able to record from. Okay, so I'll talk about two examples. And the first one actually comes all the way back from Dr. Prerau's PhD dissertation, trying to understand differential patterns of activity in the hippocampus, in particular in the hippocampus of rodents. There may be many people in the audience who may know more about hippocampus than I do, but I'll just kind of briefly state that the hippocampus is an important structure in the medial temporal lobe that's fundamental for memory and for learning. There's a famous example that maybe many of you are familiar with, a patient named H.M. Henry Molson, who had large sections of his medial temporal lobe removed bilaterally, which led to a type of anterograde amnesia where most of his long-term memory was preserved, but he was unable to form new kind of short-term memories. And after about 5 minutes of talking, he'd be able to have a conversation with somebody, say for about 5 minutes, and everything would seem coherent, but then would completely lose his memory of the past earlier conversation and would continuously reintroduce himself to you. So it's important in learning, it's important in decision-making in memory tasks, and in particular in rodents, it's fundamentally important in learning how to navigate new environments. Here I'm showing the hippocampi bilaterally in a human. In a rat, you can see the hippocampi occupy a larger fraction of the brain, which just speaks potentially of the importance of spatial navigation and learning. And this was a classic finding from 2000 from Emma Wood, that these neurons, which were classically known to relate to where an animal is in the environment, also had a property where they would fire differentially in different contexts. So here's maybe a quick movie showing an example of that. Hopefully people can hear the actual pops, the spikes from a single neuron here as well. And so if you can, what you might notice is that when the animal runs down, when it's running in the middle of the maze, you can hear a bunch of these pops, but there's a difference between when the animal is about to make a right turn and about to make a left turn. Before a right turn, you hear very few events. Before a left turn, you hear many, many more events. So this is a classic example where in this case, all you need to do is count the number of events or look at the rate of events, and you can predict whether the animal will make a right or a left turn. Of course, real data is often much messier than the videos that are shown to highlight a phenomenon. So here's an example of a more typical neuron splitter cell in the system, where on the left is a number of trials where the red line represents individual events as the animal moved down that center well before a left turn. And on the right is the same type of data, but the pattern of events before the animal takes a right turn. And if you just count the number of events before left turns and the number of events before right turns, in this example, there isn't much difference. Certainly not enough just by looking at the number of events to say with confidence that there's a significant difference between left turns and right turns. However, you may notice that it does look like there are different patterns of events preceding left turns and right turns in a case like this. So the question is, how can you determine whether there is a significant difference between left turns and right turns? Or maybe even more to the point, can you say with confidence, if I give you a new trial and the animal hasn't yet turned left or right, whether based on this pattern of data, the animal is about to make a left turn and about to make a right turn? Now, typically when I give this talk, I have people vote on what they think, whether this represents a trial from a left turn or a right turn. I'm not sure if there's a means to vote here.
You can ask people to raise hands. If they
Oh yeah, so I can maybe ask people to raise hands. Let me see if I can see the hands. I can't quite see the hands here, but maybe let's take a quick vote, and maybe someone who can see the hands, or maybe I'll see if I can do it. So how many people think that that this pattern right here corresponds to the left turn? So everyone raised their hands. Let me see if I can see the hands.
About 30%.
About 30%. How many people think it's a right turn? Okay, how many is that?
I would say, well, now people brought it down. I'd say about 50%. So maybe we have a 20% undecided.
And yeah, so that leaves maybe 20% who maybe either aren't paying attention or aren't voting, or just don't have enough confidence about one or the other. And this is typical, that I get very mixed responses where it's not clear whether this is coming from the left or right. However, as I mentioned, we can write down a point process model that describes the probability of that particular pattern of events as a function of the inputs, in this case, where the animal is, as well as whether the animal is about to make a left or a right turn. And we can build this type of point process model that characterizes at every moment the probability of seeing an event at each time, given the history, to allow ourselves to model patterns of events. And I'll just mention very quickly, the class of models we use writes down this instantaneous rate, this conditional intensity, as a function of the animal's position x of t. Here we've linearized the position on this T-shaped maze to go from negative 800 to 800, according to this kind of color code that you're seeing right here, where right turns represent positive values, left turns represent negative values. And we use what's called, and I'll talk about a little bit later, a spline basis function to relate the instantaneous rate of events to position and to direction in this case. And as I said, once we write down this conditional intensity function, the form of the likelihood is completely defined. We already know what that is. We don't have to develop any more theory. And we can use that in combination with Bayes' rule to write down a probability distribution that the context is one of left or right as a function of the pattern of spikes that we've observed. We can do this either for an individual neuron or for a population of neurons as a whole. What I'm going to show here is just for one single neuron. And this is a visualization that Dr. Prerau actually developed, which shows the distribution of this instantaneous rate, this conditional intensity, as a function of all the left turns and all the right turns, shows this particular pattern of events, and then shows this decode moment by moment. And what you can see is that not only do we end up at the end with a lot of confidence here in blue, this is the probability of a left turn, we're 90% certain that this pattern actually came from a left turn. We actually see ourselves becoming more and more confident. In particular, this first spike was much more akin to what happens on right turns. So we become much more certain about a right turn based on these first, there's actually two spikes here. But then because the rate tends to be lower during left turns, we become increasingly confident that it's actually a left turn as we have a long period here with no events. And so now you can see that the temporal patterning of spikes is playing a major role. And then these late events here, like this one, is much more indicative of left turns and also increases our confidence quite a bit. So in a case like this, by not just caring about the number of events, but by the patterning events, we end up very confident. So for the 30% of you who said left, you are correct. You should be about 90% confidence based on this long period without events here and these a few events at the end here, that this actually corresponds to a left turn. Okay, so that's the first example. I'll mention, and I'll just mention some important lessons from this case study. The primary one is that patterns of events can provide a lot more information than just looking at average spike rates over an entire trial there. And once we built a model, we got for free all of the mathematical tools for fitting those models, for decoding, in this case, left and right from those models, and something that I didn't show, assessing how well the models are actually describing the data. And this was an example where the pattern events were at least difficult to distinguish by eye, that not everybody could immediately see that it was a left turn, but we could use the model structure to actually understand what was contributing information moment by moment. Okay, I'll maybe go through briefly another example. This example is also not in the brain, or is not in the brain. This relates to the stomach of a crab, the somatogastric ganglion. And it relates to an important question in general neural data analysis, which is understanding relationships between different events, or understanding relationships between neurons, between nodes, that is functional connectivity, networks of connected point processes. And there's a common approach that you typically don't learn in an intro stats class, but will often learn in a time series analysis class or higher level stats class, known as Granger Causality, which is a way to try to detect statistically whether or not data from a particular source is informative or is causal in a certain sense of data from another source. This is statistical causality, not physical causality. And it's really about whether or not using a simple linear model, you can better predict the data at one source by using the past history of data at another source. And the reason I bring up this example is because this is one of the statistical tools that people often learn as kind of a recipe. Step by step, this is what you do, then this, then this. And often what happens there is you leave behind a lot of very valuable statistical tools for determining how well the analysis is likely to do. And this, this whole analysis was brought up based on an analysis in 2011 by Tilman Kaspersky and Eve Marder's group at Brandeis, where they basically used this Granger causality method to try to understand a network of neurons whose anatomy had already been well studied. So this is 3 neurons, the lateral pyloric, pyloric, and pyloric dilator neuron in the somatogastric ganglion, which lead to this, this oscillatory pattern, this rhythmic pattern, where the lateral pyloric will fire, then the pyloric dilator, then a period of quiescence, and then it'll repeat. And here we know exactly anatomically how this works. But when you use the kind of basic recipe Granger causality model, you end up with something that repeats the phenomenology. You see blue leads to green, leads to purple, but doesn't at all match the actual physical, underlying physical process that's generating the data. And so they concluded in 2011 that these causality methods just don't work. And so this is a fairly complicated flow chart showing this Granger causality approach. And I won't go through it in detail. I'll just mention that the first step here is to take the event data, and in order not to deal with the actual event structure, to first smooth it in time, to take bins of time and compute rates in those bins of time, and then to use that smooth data to fit linear, basic linear models. And along the way, there are a whole bunch of decisions that have to be made, like how much you smooth, and what's going to constitute a significant event. And you end up with p-values that relate to the relationships between these without, in some cases, much of an understanding for what those p-values mean in terms of the actual influence one neuron is having on another. And what we showed is if you actually use the goodness of fit tools that come with this Granger causality method, you can see almost immediately that you're not fitting the data well. If you actually look at the parameters, the model parameters that come out of this, they make almost no sense. You have, for example, here, the influence of one neuron on another at 200 milliseconds is positive, but at 205 milliseconds is very negative. That's almost certainly not actually true. And if you actually look at this model and try to use it to generate new data, so here for the first 500 milliseconds, we're just using the past data to predict the future, and things seem to work. Now, if we let the model run on its own after 500 milliseconds, it almost immediately explodes. And so our contention was, if in this paper they had used these goodness of fit tools, they would have seen right away that this model wasn't describing the data well. And what we did is we just refined the model. We used basic statistical tools to say, instead of using smooth activity, instead of using a linear structure, and instead of just using a statistical significance measure, we actually used the point process data, we allowed for nonlinear relationships, and we cared about physiological significance. Then we end up with a model structure that looks quite similar to this Grainger method. And maybe I'll just show the results. So here's the Granger causality model. Here's our point process model. You end up now with inferences shown here that almost exactly match, in this case, the true anatomy. The coupling strength you observe between these neurons matches the coupling strength that you observe physiologically, with the smallest and least significant one here, the pyloric to the pyloric dilator, being the one where there actually isn't anatomical connectivity. And if you do the same thing as before, you actually assess the goodness of fit and say, try to use this point process model to generate new data. It continues to generate the new data and does so for as long as you continue to generate data. So fundamentally, the lessons here are to use modeling methods that really reflect the statistical structure in the data, and to take advantage of the statistical tools that you have to assess the quality of the models and to make inferences about actual features that you care about in those models. And this relates to a very famous quote from a famous statistician, George Box, which is that all models are wrong, but some are useful. The tools that we have are exactly those tools that help us determine which probability models are going to be useful for making inferences. Okay, so those were the examples I wanted to focus on from neuroscience, but now I want to take the lessons we've learned from those examples and move towards, or use them to move towards understanding some sleep disordered breathing data, and move towards this idea of an instantaneous moment by moment AHI, apnea-hypopnea index. And this really gets at the same ideas that I've been talking about for the neuroscience data, that what we're trying to capture is fundamentally some kind of dynamic physiological process. In this case, for sleep disordered breathing, it may have to do with some dynamic neural processes, as well as physical processes related to breathing itself. But my understanding is often the way researchers look at this, or at least often the way clinicians try to devise therapies for dealing with apnea and disordered breathing, is by just measuring certain descriptive statistics, things like the AHI or the central apnea index, and to treat that statistic itself as the diagnostic factor. And it's important to realize, as we said before, that you have a physiological process. Therefore, anything that you use to describe that physiological process is inherently expressing some kind of model of that process. And the same is true when you look at, say, the AHI. So the AHI, my understanding is it typically comes from just looking at the total number of respiratory events over the total sleep time, and not caring at all about the temporal dynamics or the patterns of events that occur. And it turns out that that's equivalent to building a model of events where that model doesn't care about when events are occurring, just the total number of events. It doesn't care about the specific pattern of events that occur. And as I mentioned, that's a class of models called a simple Poisson process. It turns out if you fit a simple Poisson process, you end up estimating a constant rate over time, which will exactly be equal to the AHI. So another way of thinking about the AHI is the output of an estimated rate when you believe that the rate is constant, isn't dynamic, and the only thing that matters is the number of events and not the temporal patterning of events. Now, this isn't to say that there's no use in looking at the AHI. In fact, you have a model now. Once you've written down that model, you get for free a whole bunch of tools surrounding that. In particular, you get tools assessing goodness of fit, so you can immediately determine whether the AHI alone is capturing the statistical structure that's present in the data. But you also have additional measures of uncertainty. So for example, you get for free, based on that modeling, a notion of confidence intervals about that AHI. And using the point process theory, you can get exact confidence intervals. So it's well known how to take Poisson data and compute a confidence interval on the rate. The form of the confidence interval is actually a little bit messy, but fairly easy to calculate. It again only depends on two things. It depends on the total number of events and the total sleep time here. But it turns out it's actually not a symmetric confidence interval. It's actually skewed. It has some right skew. So the AHI won't be directly in the center. It'll be closer to the lower bound here. If this is too complicated, you can also show that for sufficient amount of time, total sleep time and number of events, you can build a fairly accurate approximate confidence interval quite easily. And this is the log of this confidence interval is symmetric and has the log of the AHI right at its center. And so there's a number of reasons this could be valuable. One reason is you could augment, say, patient reports that my understanding is typically now just focus on a single rate and just add to it almost immediately a notion of uncertainty about that rate, which would potentially allow clinicians to understand the potential variability that in the statistic they're using to make decisions from. You could also predict things like what might be the result if you had a patient come back for another night in the sleep lab. You could get a sense of a prediction interval for the range of AHI values you might obtain in a subsequent experiment. Okay, and you can also do the same thing for other descriptive statistics, the central apnea index as well. And extending that further, you can, instead of defining this notion of mild, moderate, or severe apnea just in terms of a single statistic, you can define ranges where you can be statistically certain of those diagnoses and ranges where you're in more of a gray area between multiple ones of these and can base your therapies based on this. So Dr. Pereira and I had a paper related to this, and he put up a really nice tutorial relating to understanding AHI uncertainty and actually an online calculator where you can put in the number of events and the total sleep time and actually estimate confidence bounds, either using this assumption that the rate is constant throughout the night or building empirical confidence bounds that go beyond that assumption and actually care about specific patterns of events. Okay, so that's the first idea, just using the fact that a point process model allows you to express uncertainty on the basic statistical features that already exist. But what if we want to extend those ideas? What if we want to build a model that can capture individual patterns of respiratory events? So in particular, we said that the way we would do that is write the conditional intensity, lambda, as a function of the things that might influence it. And in particular, we chose three categories of variables here. Two of them, because my understanding is clinicians are often interested in relationships or how these factors influence the rate of respiratory events. Things like the sleep position, whether the subject was in a supine or non-supine position. Things related to the sleep stage, so moment by moment, which sleep stage the subject is in. And in this case, we have parameters beta here and just indicator functions, in this case for position, that indicate at each time whether the subject is supine or non-supine. And in this case, at each time, whether the subject is in each of the various sleep stages we're examining. But then in order to estimate the influence of patterns of events, we've also incorporated the past history. So if you take the respiratory event chain and you just shift it over in time, you just look at what happened 5 seconds ago, and then you look at the pattern, what happened 10 seconds ago, you can get a sequence of event patterns that represent the pattern history. And you can build those all into this model simultaneously. And so we took this class of models here and fit these parameters for a number of subjects in this MESA data set, actually nearly 1,000 subjects, all of whom had AHI levels above 15. And we examined the distribution of parameters and the impact of the patterns of events and how that led to changes in the predictability of events across all of that population. And so let me just show you a few examples of what we found. So here's just one subject, just looking at about 30 minutes or so, maybe a little bit longer of a night's sleep, where you can see the actual respiratory event chain, you can see the position, in this case changing once, you can see a few changes in sleep stage, and you can look at the estimated AHI as a function of each of these models. So for our AHI model, there's just a constant rate. And here I'm writing on the y-axis the probability of seeing an event in the next second based on the model. So how confident that model is. And because the rate is constant here, that probability, you can't be very certain moment by moment. It's never changing. And you have about a 0.5% chance of seeing an event in each next time step. If you were to express your confidence, that's how confident you could be. If we just add sleep position and sleep stage, then that probability increases by a factor, an order of magnitude. So going from, say, supine to non-supine takes you from about 3% chance to about 0.03% chance. So you have a dynamic range now, and you're becoming more confident in certain positions and stages of when these events are going to happen. But once you start adding history in and modeling patterns of events, the predictability increases another order of magnitude. And so what you can see here for these individual patterns of events that we see, that these instantaneous rates are growing much, much faster. In fact, they're going up another order of magnitude. So here, say late in this burst of events here, you can be up to say 30% confident that in the very next second, there's going to be a respiratory event. And so by including these factors here, we've increased the predictability of events by about 2 orders of magnitude. Similarly, what we can see by looking at multiple subjects in the population is that even subjects with very similar static AHI levels have very different patterns of behavior of events. And so this is just 4 subjects from this population of 1,000 who all have nearly identical AHIs of about 30. But here what I'm showing is the temporal modulation. So how does an event in the past, in this case 150 milliseconds, influence the probability of an event going forward? And what you can see, there are some similarities among all the subjects. So immediately after a respiratory event, there's a period here where we multiply whatever the rate is by a value close to 0. We can call that, say, a refractory period, where more events are much less likely to occur. We also have a period indicated in each of these in green, where after an event, you're much more likely to see another event than you are, say, much further in time. So in cases here, say for this subject, after about 35 seconds after a previous event, you're about 10 times as likely to see another event. And then eventually for all of these subjects, these values go back to 1. So a multiplier of 1 then means no change in the rate. What you can see is that the pattern of, say, refractoriness and of excitability of these events is quite different from these subjects of the same AHI. In this case, you have a very large increase in the probability of an event very quickly, after about 30 seconds. In a case like this, you have a much subtler increase in the probability of an event that's delayed quite a bit. And you can see some of these peaks are much sharper and some are much broader as well. And so this suggests that there may be different clusters of patterns among different subjects. And we may be interested in exploring how those patterns might relate to other clinical factors, or maybe the potential for therapeutic benefit. Okay, and so just to maybe show a quick summary over the entire population. So this is showing these parameter estimates over all 1,000 subjects. By looking over all 1,000 subjects, you can see certain patterns emerge here. Here we're looking at the beta parameters, beta S, for N1, N2, N3, and REM sleep over all 1,000 subjects. What you can see, I think not surprising, the rates tend to decrease as you go to deeper levels of sleep and increase for REM. But one thing that might not be as obvious is that you have this kind of skewed distribution where you have tails that can get quite large for each one of these sleep stages. You also have a multiplier for non-supine versus supine sleep, where the vast majority is less than 1, suggesting a reduction in respiratory events during non-supine sleep. And then you have different distributions of parameters related to things like this refractory period, the time at which you have a peak excitability of future events, and maybe even different patterns in the widths of this increase in probability of an event. You can also use tools like this to figure out how much information from the past is actually contributing to model fit. And in this case, this survival curve shows for all 1,000 subjects, nearly all of them have patterns of activity that are informative going back at least 1 minute, but almost none of them have patterns of activity that are still informative if you go back 2.5 minutes. So we can tell that between 1 minute and 2.5 minutes is kind of the sweet spot across the entire population for predicting patterns of events. Okay, and then just the final thing, as I mentioned, once we build these models, we get for free these tools for assessing goodness of fit. And there are multiple techniques we have for assessing. One that I mentioned briefly at the beginning are these KS statistics, where a smaller KS statistic indicates increasing goodness of fit. And here we can see a progression from a model that assumes a constant rate of events to one that incorporates position and stage to one that incorporates everything. So this is the distribution of those statistics over the population. Or we can look at things like variance or deviance explained over a constant model. And we can again see this progression of increasingly explaining more and more of the variability or deviance in the data as we add first position and stage, and finally the actual history that allows us to analyze patterns of events. Okay, that's fundamentally what I wanted to say. The kind of take-home points, I think I've belabored quite a bit, but the kind of summary is that these point process tools are very powerful, that they give you for free a whole bunch of tools for evaluating uncertainty and measuring goodness of fit. And we've seen major advances in fields like seismology, like neuroscience, by developing these methods. And we feel that it's likely that there are many ranges of applications of these types of models and methods to various classes of data in sleep research. In particular, there are a number of directions that we've been thinking of for taking this type of research further, where other people might also be interested. In particular, trying to identify whether there are discrete classes of patterns of respiratory events, and if there are, what they might predict in terms of the either mechanism generating those events or the therapeutic outcomes for various therapies. One idea that might be worth exploring is whether there's potential for therapies that are based on the increase in predictability of events that come from these models. So maybe things like intermittent CPAP, where the amount of air that's being blown through changes as a function of whether or not events are likely to occur in the next period or so. And then finally, I've focused here only on sleep disordered breathing and on respiratory events. You can think of many other types of events, things like sleep spindles or sleep stage transitions that might also be well fit using these types of point process models. Okay, so that's it. I just maybe want to thank all the people who actually built the models that I've described here, including Dr. Prerau, who did most of the analyses on the hippocampal data that I showed you, and the data collaborators and methods development, again, including Dr. Prerau, who has really helped with the development of the models for those sleep disordered breathing applications. And then, of course, all the other labs and groups that helped with the modeling and with the data, and the various institutes that helped support all this work.